right, we'll start off this evening, and I'm going to begin with lesson 11. And this lesson has to do with genes and proteins. And so really what we do, we call this the central dogma of biology. And what that means is, for every gene, the gene is sort of the code or the instructions, and there is some product for every gene. And so if we start here and we say, all right, on the DNA, at the DNA level, right, the DNA can be broken up into little segments that are called genes. So the gene is on the DNA, or we could say, you know, the chromosomes, right, are made up of DNA, so genes are found on the chromosomes. And so these are like the instructions, okay? But ultimately, okay, I'm going to leave a little spot here in the middle, but ultimately the product of that gene is a protein. And the gene itself, the instructions for the gene, is what we would consider our genotype, okay? And the protein product that this gene codes for is what we would consider the physical appearance or the phenotype. Now, the central dogma essentially says, in the cell, how does the cell turn one of these genes on, right? How does the protein product of that gene actually get made in the cell? That's what we're going to talk about in this lesson. So it turns out that there's an intermediary, okay, so there's a middleman in this process, and the middleman is a special type of RNA called mRNA or messenger RNA. And so this is, I've drawn two arrows there. So in other words, the first arrow gets us from the DNA to the mRNA, and the second arrow gets us from the mRNA to the protein. And so this first process is called transcription. And the second part is called translation. Now, for prokaryotic cells, they don't have a nucleus, okay? So it's a little bit different for them. But for eukaryotic cells, the transcription part has to occur in the nucleus because that's where the DNA is, right? The DNA is always found in the nucleus. So the DNA to RNA portion is happening in the nucleus. But the mRNA to protein portion is happening outside of the nucleus, and it actually happens in the cytoplasm, okay, or on the rough ER, either one, okay? But the key is the spot, okay, or the machinery that's helping this to occur is called the ribosome. So whether it's a ribosome that's free in the cytoplasm or a ribosome that's found in the rough ER, that's the place where the protein production is being carried out from the mRNA. Now, I want us to talk in a little bit more detail. Let me just get us a clear. I want us to talk a little more detail first. Let's start with transcription. Okay, so for transcription, we're starting with a DNA template. And remember that DNA in the cell is double-stranded, right? We have two strands of DNA that are bound together. So if we just sort of focused in to a segment of DNA, okay, let me see here. Okay, so let's say that one strand of DNA has this particular nucleotide sequence. So it's much longer than that, right? It continues on, okay? But we're just focusing in on the actual nucleotide sequence of one portion. Now that would mean that the other strand, Okay, I'm gonna draw it in a little bit different color here. That since it's double-stranded, right, the other strand we could determine what the sequence is because there's a certain way that they pair. So A always pairs with T. Okay, and this is what our double-stranded DNA would look like. But when transcription occurs, unlike replication, so when we talked about DNA replication, both strands got replicated, right? The DNA polymerase enzyme came in, and a copy was made of each strand, and so each strand had the old and the new copy. But with transcription, that's not what happens. With transcription, there's only one of these double-stranded DNA sequences that's used as what we call the template strand. Okay, so let's say that in blue, okay, that this is going to serve as our template strand. So this is the one that is actually going to be transcribed into mRNA. So I'm just going to put a line through this red sequence because, in other words, that sequence is there, but when the DNA gets unzipped, only one of these strands is going to be read as the template, okay? And we're going to say it's the blue strand is representative of the template. Now this is happening in the nucleus, and there's an enzyme that performs this function, okay? And in DNA replication, the enzyme was called DNA polymerase, okay? In this process, the enzyme is called RNA polymerase. Okay, so it was called DNA polymerase because in DNA replication it was a DNA strand that's being produced, but what we're producing here in transcription, what we're mimicking is an RNA strand, right? It's a specific type of RNA called mRNA, so the enzyme is called RNA polymerase. So the enzyme we're talking about RNA, so remember there are a couple of differences between RNA and DNA. One of those is the type of sugar. Also, RNA is typically single-stranded, not double-stranded. But the other thing is, in RNA, we have the nucleotides A, C, G, but we don't have T. Instead, we have U. So for everywhere that there would normally be a T in DNA, it's going to be a U in RNA. So the transcription of this, that this enzyme is carrying out, so when it reads this A, okay, instead of adding a T, right, that would be the normal base pair. Instead, it's going to add a U there, okay? And then it reads a T, so it adds an A, and this process continues. Okay, so that would be the mRNA, okay? M standing for messenger RNA. Why messenger? Because it's carrying the message that's found in the nucleus on the DNA out to the ribosome where it can be then translated into protein. Okay, so this would be our messenger RNA, but something has to happen to the messenger RNA. Okay, it's not considered mature at the point it can't leave the nucleus. And so one thing that has to happen is it gets a poly A tail, okay, added. So lots of adenine nucleotides get added to the end. It also gets a guanine cap added to the opposite end. And then there are sections of the mRNA that get spliced out. So let's say that I'm going to underline certain sections. So this is going to be considered an exon, okay? And this is considered an exon. The part that's not underlined, let's say that is considered an intron. So that portion will actually get spliced out, okay? So in other words, this portion would get cut out and the exons would get put together, okay? That is called processing. The tail being added, the cap, and the introns being spliced out. That's mRNA processing, and you could say that that's how the mRNA is mature, so that it can now leave the nucleus and travel out and to the ribosome where translation will occur. I just want to show you a couple things real quick. Okay, so if you imagine that in these regions of color, okay, so all of these would have nucleotides, right? So this might be a G, right, A, U, so on and so forth, okay? So the introns are those that are going to get spliced out, okay? And then the exons are put together. Notice the poly A tail has been added, and the 5' cap has also been added. Okay, so this would be a mature mRNA that would be ready to move on for translation. So let's look at another figure really quickly. I'm going to talk you through this. So this represents the mRNA, okay? So this has got only the cap, the exons, and the tail. Okay, this is ready for translation. Okay, so remember the first step was DNA to RNA, that's transcription, and then RNA to protein is translation. Now, this occurs on the ribosome, and the way that these come together, okay, the ribosome has two different subunits. It has a small subunit and a large subunit. And the ribosome is made up of a special type of RNA called rRNA. Whoops. Okay, rRNA, and also some protein. So that's what makes up the ribosome. So this little piece here represents the small subunit of the ribosome, and then this much larger piece represents the large ribosomal subunit. So you see that that several things have to come together in order for translation to begin. And so what we see here is we have the mRNA, right? This is what is going to be read by the ribosome. These are the instructions that are carried from the DNA. We have that. We have the small subunit that joined this transcript. And then this other member here that we see is a tRNA. So you can think of T standing for transfer. So in this figure, in this top part, we have three types of RNA. The mRNA is the message, okay? The rRNA helps make up the ribosome, and the tRNA, which is the transfer RNA, is responsible for carrying the correct amino acid, okay? Proteins are made of amino acids. So you put multiple amino acids together, that's going to be the protein product. So we'll talk about that in detail in just a second. But the last member to the party then is the large ribosomal subunit. So we see that here, okay? And this is what happens during translation. First of all, the ribosome essentially is reading the nucleotides on the mRNA in sets of 3, okay? So that's the first set of 3 AUG, and that's actually called the start codon. Okay, but each group of 3 nucleotides makes up a codon. And every group of 3, every codon then, codes for 1 amino acid. So 3 nucleotides for 1 amino acid. Now I'm going to take us away from here for just a second because I want us to look at this genetic code chart right here. So this is showing all of the possible codons. So if you take all 4 nucleotides, A, C, G, and A, C, G, and U, and you combine those in every possible way that you can combine those 4, right, in their sets of 3, you come up with, I believe it's about 64 total combinations. And so you see them all here. Okay, what you also see is what particular amino acid that's coded for by that particular codon. So for example, AUG, the first one that we just looked at, okay, AUG you find right here. So to use this genetic code chart, first thing you do is find the first letter. Okay, so that's A. That puts us in this row, okay? Then you find the second letter, which is U, okay? So then that means, all right, we must be in this column, therefore we're isolated right to this box. Then you find the third letter within that box, so there's our AUG. That is our start codon. Now, a couple other things I want to point out to you besides it being 3 nucleotides for every 1 amino acid, and that is that it's redundant, right? Or degenerate, meaning there are multiple codons that code for the same amino acid. So let's take this box for example. Okay? The amino acid serine has all these combinations of codons that will always code for the amino acid serine. Okay, and there's 2 more down here that also code for the amino acid serine. Okay, now when you think about it, part of that is so that if there is an error, right, and it was supposed to read UCC, but there was a mutation that occurred and one of those C's became a U, so now it's UCU. Even though there was a mutation in the genetic code, when we see the protein product that's produced or the amino acid that's coded for, it doesn't change. Therefore, the actual protein itself is going to be unharmed or unchanged. Okay? This genetic code is pretty much universal from bacteria, animals, plants, humans. It's the same sequence of codons that code for these particular amino acids. Now, the other thing I want to point out to you are these that are in bold, okay? These codons are what we call stop codons, and what that will mean is when the ribosome gets to that point and there is a stop codon, essentially it releases the mRNA, it releases the growing protein because that is saying we are finished, right? That's the end of the protein. Okay, so we'll go back here. Now that we recognize that these are in codons, okay, so remember, this is the mRNA, and we're only seeing a few codons, right? But all of this mRNA would be made up of codons. So this first codon is AUG. That is, remember, that is the message, right, that was coded for by the DNA. Okay, the instructions for what this protein should look like were originally coded for by the DNA. So it's been essentially copied, right, into mRNA. And now the tRNAs are going to do their job, and that is to bring in the correct amino acid for that codon. And the way that this works, the tRNAs actually, essentially, you can think of them as having 2 ends, right? This whole molecule right here is a tRNA, and this whole molecule here is a tRNA. One of them is the transfer or tRNA or transfer RNA from methionine, and one is for phenylalanine. Now, one end of the tRNA is going to be complementary to a codon on the mRNA, meaning just like DNA base pairs A with T, okay, it's the same except remember with RNA there is no T, so it's A with U, C with G. So this portion of the tRNA that's complementary to the codon is called the anticodon. The other end of the tRNA is attached to the appropriate amino acid. So in this case, methionine is the correct amino acid. So the next tRNA that's coming in, AAG, is complementary to this next codon, and it has the appropriate amino acid attached, phenylalanine. Okay, so notice that there are a couple of binding sites for tRNA molecules on the ribosome. So we have one here, okay, we have another one here. These are called the P or peptidyl binding site and the A binding site. Now, once they are both bound, Okay, what's going to happen is there's going to be a bond that's formed between the two amino acids. So notice up here in this figure, okay, these amino acids are in no way connected to one another. Methionine and phenylalanine are not bonded together. But when they become, when this one comes in and bonds to this site, there will be a covalent bond that occurs between methionine and phenylalanine, okay? It's a special type of covalent bond that is going to occur between every single amino acid in this growing peptide chain or growing protein chain, okay? And because it's a peptide or protein chain, it's called a peptide bond. Now, as this occurs, so now we see, right, that there is a bond between these two, and the tRNA will eventually be ejected, okay, out of the ribosome, but it will leave behind its amino acid, okay? So if you imagine as the ribosome shifts this direction, okay, to the next codon, then there's going to be a bond that forms between this amino acid, arginine, and phenylalanine. This portion of the tRNA will be ejected, but all of the growing, right, amino acid, the growing peptide chain is going to stay attached, right? So these numbers of peptide bonds just keep growing as each new tRNA brings in a new amino acid. Now, I keep saying amino acids and proteins. The protein is the chain of amino acids. So when the ribosome gets to the stop codon, that means that is the end. That's the last amino acid that's needed for that protein. So that long chain of amino acids, okay, that does become the protein product. The protein then becomes folded in a correct way and will serve as a functional protein. Okay, now I do want to show you, that was, let's see. Okay, so just an overview, right, of the whole process, transcription and translation. Remember that the transcription portion, right, at least in a eukaryotic cell, is going to happen in the nucleus. Now, in a prokaryotic cell, there is no nucleus. Okay, the DNA is free in the cell. But you see both strands, so shown in blue, right, are both of the double-stranded DNA, okay, that's found in the nucleus. Okay, so those strands have to separate so that the enzyme that makes the mRNA copy from the DNA template can make a copy from only one of those strands. Remember, only one of those is going to serve as the template. Okay, so in this case, this strand is serving as the template. And just like DNA polymerase can read DNA in only one direction, RNA polymerase can read DNA in only one direction. It reads the template in the 3' to 5' direction. Therefore, the new mRNA strand is produced from the 5' to the 3' direction. And remember that the mRNA has U's, right, instead of T's because RNA doesn't have thymine. And then you see the processing of the mRNA. So the primary mRNA transcript has both exons and introns. So those introns have to be spliced out. A cap is added. A poly-A tail is added. Then that mature mRNA can exit the nucleus, where the ribosome, right, the small and large ribosomal subunits will come together along with the mRNA transcript, and the tRNAs will bring in the correct amino acids for this growing polypeptide chain. Now, after this test, we will spend some time talking about how is this regulated, right? How are genes turned on and turned off and all of that good stuff. But for now, that's just the nuts and bolts of how transcription and translation actually occur in the cell. Now we'll switch gears and we will talk a little bit about genetics, Mendel and genetics. Okay. All right, so Mendel was such an interesting man, okay? Father of genetics, wasn't even recognized for all of his contributions until after his death, but he crossed thousands of pea plants in order to establish some basic understanding of genetics, okay? So his model organism, the one he used to demonstrate the traits that he was interested in, was the pea plant. And one of the things that he noticed right off the bat was that there were such a thing as dominant and recessive traits. Okay. And what did he mean, and what do I mean by that? Well, he said that a dominant trait, if it's present, can mask the recessive form of the trait. And the recessive form can be masked if there is a dominant allele present. So for example, let's think about some of the traits for peas. So for example, one dominant trait would be round seeds, and the recessive form would be wrinkled seeds. And so he noticed that if you crossed a homozygous round pea plant with a homozygous wrinkled pea plant, all of the offspring were going to be round. Okay, they were all round. But then if you cross those offspring together in the next generation, you would actually see some round and some wrinkled. So what he realized was there's something that is being passed from one generation to the next that you can't see, right? And he called that a trait, which we now know is a gene that's being passed. So if we use big R to represent the round form and little r to represent the wrinkled form, then we can cross. So we're going to say we have two parents, okay? So one parent is homozygous dominant, okay? Now let me just do some more terms here. So we have homozygous, okay? This is two of the same alleles, okay? Alleles just means a form of the gene. Heterozygous then would be two different forms. Heterozygous, okay? Two different forms of the gene or of the allele. So homozygous dominant, that means two of the same and it's the dominant allele. Homozygous recessive, homozygous is two of the same and they happen to both be recessive. Okay, that's the parent generation. So he's saying if you crossed homozygous dominant round pea plant with a homozygous recessive wrinkled pea plant, okay, the F1 or the first filial generation, so in other words, the first offspring from that, would look like this. So they would inherit one allele from this parent and one allele from this parent, but there's no, they are definitely going to inherit the dominant allele from this one, and they're definitely going to inherit the recessive allele from this one, so there's really only one genotype, right, for these offspring to be big R, little r. And what do they look like? Well, they have round seeds because they have one dominant form of the trait which masks or covers up the other one. Okay, but what he said is, but when you take the F1 generation and you either allow it to self-pollinate or you cross two together, you then are able to see both round and wrinkled seeds. So if we take F1, so we say, okay, we're going to cross big R, little r with big R, little r, and by the way, this would be what we would call a monohybrid, mono meaning one trait, hybrid meaning one of each, right? One dominant, one recessive, monohybrid cross. Now we're going to need to set up a Punnett square in order to do this cross. Okay, so with a one trait cross, your Punnett square needs to have 4 boxes. We're going to take, let's say we're going to take this parent, and we're going to figure out what possible gametes can this parent produce. So a gamete, which is either sperm or egg, has gone through meiosis, right? So the gamete has half of the genetic material. So the gamete is either going to get this big R, or it's going to get the little r, but it will only get one. So this represents one gamete, and this represents the other gamete. This parent, pardon me, this parent has the same genotype, so one gamete will have this one gamete will have this one. It makes no difference what order you write these in. Now the Punnett square, inside the Punnett square represents the possible offspring that these parents can have. So above and to the side, right, these are the gametes for the parents. In the boxes are possible offspring. So then we're just going to carry this R across and carry this R down. Okay, so big R, big R, big R, big R. Then we'll do the same thing with the little r. This little r will carry down, little r, and we'll carry this one across. Okay, so that doesn't mean that they'll exactly have 4 offspring and they'll exactly be this genotype. That means probability-wise, this is the likelihood, right? This is the potential genotypes they can have, and here's the likely probability. Just like if I flip a coin, I have 50% chance I'm going to get heads, 50% chance I'm going to get tails, but I have 2 possibilities, heads or tails. So in this case, we can report this, okay, based on genotype. So we can say, what is the genotypic ratio of this cross? Okay, well, the genotypic ratio would be 1 big R to 2 big R to 1 little r. Okay, or you could say 25% big R, 50% big R, and 25% little r, right? Those are, they have the same meanings, or 1/4, 1/2, 1/4, all that. It's just different ways to say the same thing. Now, what if we look at the phenotypic ratio? So the genotypic ratio is what the gene instructions are for, right? Actually at the gene level. So we're using letters like R to represent a form of the gene, right? So that would represent a certain gene sequence or allele for that gene. The phenotypic ratio means what is the actual physical appearance. So in this case, does it have a round seed or does it have a wrinkled seed? So phenotypically, it's 3 to 1. So 3 round, you usually will report the dominant phenotype first. So the dominant phenotype is round, so 3 to 1. Or 3 out of 4 would be round and 1/4 would be, or 75% to 25%. Again, they all mean the same thing, right? Whether you use percentage, fractions, or ratio. Okay. So let's talk about another type. So this would be what we would call complete dominance, right? Where one trait completely masks another trait. Let's try one more example with complete dominance, and then we'll move on. Okay. So let's do a human trait. Okay. So let's talk about albinism. Okay. Is a recessive it's recessive, let's see, it has a recessive, let's say it's a recessive trait. Okay, so when I say it's a recessive trait, what I mean is in order to be albino, you have to have both copies of the recessive allele, meaning the normal allele means you are not an albino. Okay, so let's just use M, okay, for melanin. So big M would be normal, right? No, little m is going to be the allele for albinism. But an individual only is albino if they inherit both of the recessive forms, right? The dominant one is going to be the normal type, and that's going to mask over the recessive if it's there at all. So let's say, what if we have two parents, okay, and both of them have a parent who was albino, okay? So let's give them a name. Okay, let's say we have Jack, and we'll have to just use Jill, okay? They didn't go up the hill, but Jack and Jill. So Jack had one normal parent, one albino parent, so did Jill. So what that means is, from the normal parent, Jack had to have inherited only the big M, right? Because, well, let's say they were homozygous normal, so they inherited that from one parent. Now, Jack has one parent who was albino. That means one of his parents had to be little m, right? If they were albino. So we know that Jack inherited one little m. Okay, Jack is not albino, which this works because if he got one normal one, he would appear normal. So Jill has the same genotype, right? Because she had one parent who was albino, which means she had to inherit one little m from that parent. So the question is, when Jack and Jill have children, what chance do they have to have a child that's albino? So we'll have to set up our Punnett square. Okay, again, we're only doing one trait, so we just need 4 boxes. So let's put Jack's gametes across the top. So we'll split them. Remember, gametes are half the genetic material, so one big M there, one little m there. Same thing for Jill, one big M, one little m. So now we just fill in the boxes. Okay, and what we find out is they have a 25% chance of having an albino child, right? 1 in 4. Okay, let's move on to something a little more complicated. Let's talk about dihybrid crosses, okay, or 2 traits. So we can look at 2 traits at once and how they're inherited as long as they're not linked on the same chromosome. So after the exam, we will talk about linked genes and cover some of that. But if 2 genes are carried on different chromosomes, okay, then the law of independent assortment tells us that it doesn't make any difference which way one chromosome lines influence the way the other chromosome lines up in meiosis, meaning they are inherited completely separately, okay? So we can treat them as such. So in other words, we can look at all possible combinations. So let's talk about another couple of human traits, okay? Let's say that widow's peak, okay, is dominant to a straight hairline. Okay, so this is okay, and this one's straight hairline. And then the other trait we'll look at is, okay, capital B would be one. There's multiple genes for eye color, but let's say the presence of a particular allele that makes brown eyes is big B, and the recessive form of that, okay, would make some kind of light eyes. We'll say blue to make it easy. Okay, so if we want to do a dihybrid cross, that means an individual is heterozygous for both traits. So heterozygous means one of each. So that would mean big W, little w, big B, little b. Okay, so if both parents have this particular genotype, that's a dihybrid. Now, you can still do the Punnett square, right? You can still do the cross if they have some other combination, like let's say one of the parents is homozygous dominant for widow's peak and homozygous recessive for blue eyes, right? It's just not called a dihybrid cross. cross in that case. Now, the first thing we have to understand, since we're talking about two traits, is our Punnett square is going to have to be quite a bit bigger, okay? We're going to have room, whoops, that doesn't look too good, for 16 boxes, okay? So again, outside the Punnett square are the gametes for each parent, and inside is the possible offspring genotypes, and then we can look at the probability, right? So in order to figure out what goes in the gametes here when we're talking about two traits, because essentially because these are not linked genes, we need all possible combinations of the way these genes can shuffle in meiosis to form different gametes. So let's take, sorry, that's supposed to be a W, my pen went crazy. And if you haven't heard of the technique called FOIL, that's what we're going to use. So it stands for first, outer, inner, and last. So if you think about this as being two traits, right, we're going to take the first for both traits, so that would be capital W, capital B. Then we're going to take the outer, so that would be the outer, this would be capital W, and the inner here would be little b. Then we're going to take the inner, so that would be little w and b. And then we're going to take the last two for each, so that would be little w and little b. And essentially what you've done with this foiling method is you've figured out what are all possible ways you can combine these together. Okay, so because both parents have the same genotype, right, we're going to do the same thing for this parent. Okay, and then comes the easy part where you just have to fill in the boxes. So you carry this one all the way across and these all the way down. And I like to group my different letters together. It just makes it easier, okay, to keep track. Okay, so you're going to fill in all of those boxes. And what you're going to realize is at the end, a dihybrid cross, so only in the case when you're crossing two dihybrids, you will always get a phenotypic ratio of 9 to 3 to 3 to 1. So out of that 16 boxes, 9 of them will have the dominant phenotype for both. So in other words, in this example, they'll have a widow's peak and brown eyes. 3 of them will have a widow's peak and blue eyes. 3 of them will have straight hairline and brown eyes. So in other words, those are one dominant form of one trait with the recessive form of the other. And the last little individual will be homozygous recessive for both traits. So straight hairline and blue eyes. So that's how you would deal with two traits at one time. Now I want to talk just a minute about some instances of inheritance that are not completely dominant. Okay, so there's two examples that I want to talk about. One is called incomplete dominance, okay, and the other one is called co-dominance. Now, when you think about incomplete dominance, what I want you to think about is in between, okay? So in other words, we have a phenotype that is in between or in the middle. So this example would be you cross a red flower, you cross a white flower, you don't get either red or white, instead you get pink. You get an in-between phenotype. For codominance, you actually get both expressed, okay? So what that would look like, you cross a red flower and you cross a white flower, and you don't get a pink flower, instead you get a flower that has parts of it that have red and parts of it that have white on the same flower or the same petal. Now, it's important in these examples that you work it out and do the Punnett square, because since you have a third phenotype, sometimes it's not obvious what the answer would be. So an example of a codominantly inherited trait is something called roan coat color in cattle. Okay, so let's say that we're going to use the superscripts, right? So let's say R1 is a cow with red coat. Okay, then we'll say R2 is a cow with white coat color. Well, if we have R1, R2, in other words, it's the heterozygote form, right? One of each. It's not red or white, it's something called roan because it expresses both red and white hairs, and it gives it this particular color. Okay? So in an example like this, if we cross two roan, like a roan cow and a roan bull, okay, still it's just one trait, right? So we only need the 4-box Punnett square. So we're going to cross two of these. So R1, R2, that's the gametes for one parent. The gametes for the other parent will be the same. Okay, so what we find out is 25% will be R1, R1, which is red. 50% will be R1, R2, which is roan. And 25% will be R2, R2, or white. So that's an example of codominance. How do you identify which one is which? Well, you're looking at in between. So like red and white make pink, or curly hair and straight hair with wavy hair in between. Okay, that would be the difference between incomplete and codominance. Okay, I'm going to move on and talk about X-linked genes, I believe. Actually, no, let's talk about blood type first, and then we'll finish up with X-linked genes. So blood type is really interesting because it is both codominant it has what we call multiple alleles. I'm going to explain this in just a minute. And it also has complete dominance. Okay, so let's talk through what do I mean about blood type. Okay, so what I mean is you probably are familiar with someone can be type A blood, someone can be type B blood, someone can be type AB blood, and lastly, someone can be type O. Now, that you also recognize blood type as, let's say, A positive, O negative, right? The positive or negative. I'm not going to mention that, but all that is an additional gene, right? That you either have the presence of that factor or not. So it works similarly. But here's what I want to talk about. Type A blood, okay, comes from an allele, okay, that makes a particular antigen on the red blood cell. So we're talking about what is occurring on the surface of the red blood cell. Okay, so if someone is type A, that means they have this particular antigen on their red blood cell. Okay, we're going to represent with a capital letter I with a superscript of a capital A. Okay, in other words, that represents the allele that codes for this A antigen, okay, on the red blood cell. Type B, okay, we're going to represent with capital I with a superscript B. Okay, and what does this represent? Well, this means this is the gene that would express this other type of antigen on the red blood cell that we're going to call the B antigen. Okay, Someone who has type AB blood, then what that means is they have to have both of those, right? They get one of these type A's and they get one type B. So we'll put that here. The only way to be AB is you have to have one of the A alleles and one of the B alleles. Now, to be type A, that's the allele that causes the A antigen. Everybody, right, has 2 chromosomes. We've got one from our mom, one from our dad, so we have 2 copies of everything. So the person can either be IA, or they can have only 1 copy, which is dominant to another allele that I'm going to represent with lowercase I. Same thing with type B. They can be IB, or they can be IB and have this other allele that's recessive, okay, that I'm going to represent with lowercase I. What does type O mean? O means the absence of A or B, right? You don't have any of those antigens on your red blood cell. The only way to be type O is to have 2 copies of this recessive, essentially non-functioning, right? It doesn't make an antigen on the type O blood cell. So codominance is going on because the A antigen and the B antigen are codominant, right? They can both be expressed, so type AB blood is showing codominance, right? It can be truly dominant because the A and the B are both dominant over the recessive form. So in other words, if it's IA little I, it's still type A blood, okay? Or if it's IBI, it's still type B blood. And it's multiple alleles because we don't just have 2, right? There's 3 choices. There's an IA allele, there's an IB allele, and then there's a little I allele. So we have a lot going on with blood typing. So let's say that a person with type AB blood, okay, let's say a woman with type AB blood has a child with a man with type O blood. Okay, so the woman is AB and the man is O. Okay, so the first thing we need to figure out is each of their individual genotypes, and this one's easy because there's really only one way to be AB. Okay, so IA, IB. And there's only one way to be O, and that is to be little I, little I. So when we set up the Punnett square, let's see, I'm gonna move it over here. Okay. Again, we're only talking about a 1 trait cross, so we only need 4 boxes. So let's put the mother's gametes here, so half the information. So one gamete can get the IA, the other gamete can get the IB, and we'll put the father's down the side, so his sperm can only get the little I. That's all he's got, right? We fill in the Punnett square. What we see is they have a 50% chance of having a child that the genotype is IAI, which means type A blood. And they have a 50% chance of having a child that's IBI, which is type B blood. So their children will not have the blood type of the children will not match the blood type of the parents. Okay, last thing I'm going to cover tonight is Xlinked traits. So for X-linked traits, that means that it's a trait that's carried on the X chromosome. So we have 22 autosomes, okay, which are not sex chromosomes, and then we have a pair of sex chromosomes. So for females, we have 2 X's, and for males, they have an XY. So with X-linked, it's very important that we identify if we're talking about a male or a female, because a female has 2 X's, right, that we have to figure out what form of the gene is on the X chromosome. But the male only has 1 X and then has another Y. So let's use, for example, hemophilia. Hemophilia. Okay. Hemophilia is a recessive disorder. Okay. So what that means is if you have one normal or good copy of the gene, you will not be affected with hemophilia. So we'll represent the hemophilia. So big H is going to be the normal, right, because it's dominant. And little h is going to represent the affected, right, or the hemophilia allele. So let's say we have, let's use Jack and Jill again. Okay, we've got Jack and we've got Jill. Okay, so Jack is normal. He is not a hemophiliac. So that's easy for us to figure out. Okay, so the first thing we need to do is we need to put Jack's sex chromosomes down. We need to put Jill's sex chromosomes down. Okay, that'll help us keep track. We don't put anything on the Y because this is not carried on the Y, it's only carried on the X. So the Y is sort of a placeholder there to tell us whether the children are going to be male or female in the Punnett square. But because we know Jack is normal, we're going to put a big H right up here with the X because we know he's not a hemophiliac. Now, Jill, on the other hand, okay, her father was a hemophiliac. She's normal, okay, but her father was a hemophiliac. So since she's normal, we know that she gets one big H here. But her father, right, we know that if her father was a hemophiliac, he had a little H here. Well, if she had inherited the Y from her father, she would not be a female, right? She would be a male. So we know that she had to inherit this affected X. So this is her genotype. She does not have hemophilia, but she carries that affected allele, so we would call her a carrier. So let's set up the Punnett square, okay, for this cross. And it's very important when you report for sex-linked disorders that you report male children versus female children because as you're going to see, it's much more predominant in an X-linked disorder to see it in the males because they don't get two X's, they only get one. So they either have the disorder or they don't. Males can't be carriers. All right, let's put Jack, okay, across the top. So we'll put his X big H here and his Y here. We'll put Jill's across the side so she has one X big H and one X little h. So if we fill in the Punnett square, all right, so we're gonna talk male children, okay, separately from female children. So if we look at the male children, that's this half over here. What we see is we have 50%, right, that are X big HY or normal, and we have 50% that are X little hy or are hemophiliacs. For females, we have 50%, okay, I'm not writing all this down because I don't have room, but 50% that are completely normal, right? They're homozygous for the normal allele, and we have 50% carriers, right, that are heterozygous. They have one good and one bad. But we have no female children that have hemophilia, whereas 50% of the male children are going to be hemophiliacs. So as you work through the X-linked problems, you'll see that red-green color blindness, male pallor baldness, these things, hemophilia that are predominantly seen more in males than females is because they're carried on the X chromosome and males only get one copy of the X chromosome. So that finishes our review tonight. As always, email me if you have any questions.